I'll try to do is give you sort of a flavor for where I see genetic metabolic diseases today and where we're going with treatments, and just sort of give you an update. But I'll also try to bring everyone to the same level, remembering that we're a very eclectic audience. I am a card-carrying geneticist, and so you're wondering, what's a geneticist doing in pharmacology or in College of Pharmacy? Well, we'll find out. But I use a lot of different pharmacological approaches to treat genetic diseases, and most of my work is in mouse models. So we'll touch a little bit on that just in a few seconds. So what I'm going to talk to you about today are what are really inherited metabolic diseases. How frequently do they occur? Because you're probably thinking, these are pretty rare things. And generally speaking, a lot of them are pretty rare. But if you look at the cumulative number, it gets a little bit more significant. How do we detect them in the laboratory? So how do you screen for newborns that might have an inborn metabolic disease? What are the traditional treatments? What have we been doing up until the last 5 or 10 years? And then what's new on the treatment landscape? And that's particularly interesting to me and to others that are interested in inherited metabolic disorders. So that's what we hope to get through in the next 20 or 25 minutes. And then I'll tie it up a little bit at the end. So inherited metabolic diseases. They were really sort of, let's go back a step. So you all remember, or at least those of you who took genetics, you remember the old Mendelian green peas, yellow peas, and that was inheritance, and people started to learn about that then from Gregor Mendel. But really the title inherited metabolic disease came about from an English physiologist by the name of Archibald Garrod, and it was coined, that term, in 1908. And that's a picture of Sir Archibald Garrod. And he was studying a variety of genetic diseases back then. And you can imagine in 1908, he didn't have the most sophisticated methodology or techniques. But he was a clever guy. And he was actually studying this disease called galactosemia, which is basically the original sort of inborn error of metabolism. It's a defect in the metabolism of the amino acid tyrosine, okay? And he was as I said, he was a very astute clinician, and he looked at the issue of inheritance. And so what do we mean by inheritance? He had a series of patients. He followed several cohorts of patients that had this debilitating sort of joint disease. And we call it in the clinical trade ochronosis. So they actually had precipitation of this compound showed here, homogentisic acid. And you won't be quizzed at the end of this, so don't worry about that. But he collected urine from these families, and actually he noticed that when the urine from some of the children sat for a while outside at room temperature, it turned black. And he coined the disease, or the title for this disease, black urine disease, okay? And it turns out that this compound, homogentisic acid, when it oxidizes, it turns black. And these kids, certain of these young patients, had huge amounts of homogentisic acid in their urine. He collected urine from the parents, let it sit out, it didn't turn black. It turned black from the urine of both males and females. And so he started to really understand the inheritance. If it was only in males, he would have known that the trait was passed from mother to son, and it's probably X-linked inheritance. The parents didn't show any evidence of the disorder, so it wasn't an autosomal dominant, because autosomal dominant diseases will invariably show up in all lineages, parents to offspring, and then again to new parents. So he coined the idea of autosomal recessive diseases. He knew it had to be recessive. And what do we mean by recessive? And that's shown here basically on the left, which is hard to see. But you have to have a carrier father and a carrier mother, so they have one abnormal copy of the gene each. And then in their offspring, there's a 1 in 4 chance for a child to receive one copy each of the bad gene, or of the defective gene, if you will. So it's a 1 in 4 chance, and following it, you could have a homozygous unaffected patient. Then you have 2 in 4 chance for carrier offspring. They each have a copy of the bad gene, just like the parents. and then you have that 1 in 4 chance of having an affected child. So he knew or understood then that human metabolic diseases follow Mendelian characteristics, just like Mendel described back with the yellow peas and the green peas. So what is the frequency that we're talking about with some of these diseases? Many of the inborn errors of metabolism are exceptionally rare, okay? So 1 in 1/4 million live births to well less than 1 in 1,000,000, 1 in 4 million perhaps. Some are much more prevalent. So phenylketonuria, which is the most common of the inherited amino acid ureas, is about 1 in 9,000 to 1 in 11 or 12,000 in terms of live births. So if you think of a state like Texas, where they have upwards of about 400,000 newborns per year, if you have a frequency or an incidence of 1 in 10,000, you're going to have 400 new cases of phenylketonuria per year, on average. Some of these inborn errors are much more common in what we call consanguineous matings, and that basically comes from the Greek, which is sanguine, which means basically blood. Consanguineous matings are basically interrelated marriages where a cousin, first cousins or second cousins or third cousins will marry. And what that basically means is that recessive genes, because you have a very small mixing pool, become much, much more frequent in consanguineous matings. So for example, examples of that would be Tay-Sachs disease, which is a debilitating disease, very bad disease, which is very common in Ashkenazism, in Ashkenazi Jewish parents. Maple syrup urine disease. So normally, the carrier frequency is very low for maple syrup urine disease. It goes to about 1 in 10 to 1 in 20 in the Mennonite population in Lancaster, Pennsylvania, where that's a very small group of individuals that marry within families. So if we look at the overall frequency total for all of the genetic diseases, inborn errors, and metabolism, it's about 1 in 3,000 live births, 1 in 2,500 to 1 in 3,000. So it's a lot more common than you would have thought. So how are inborn errors of metabolism detected today versus yesterday? And I thought I'd show this slide and then this slide. So what I'm showing over here, and it's hard to see, is the original what we call Guthrie plate. And Robert Guthrie in the 1960s really invented the whole idea of newborn screening for genetic diseases, and specifically for phenylketonuria. And so back in the dark ages, but still some labs probably even use this today, this is basically a screening plate, and what you have embedded in this agar gel are bacteria. And these bacteria basically need phenylalanine to grow. So they're basically what we call auxotrophs. They're genetically manipulated so that they have to have phenylalanine to grow. And basically what's shown here are increasing amounts of phenylalanine in a spiked blood spot. So all newborns, when in the nursery, a day of life 1 at about 24 hours, they'll get their heel prick, they'll collect blood on a newborn screening card. It gets sent off to the screening laboratory, and they get screened for a number of genetic diseases. We'll talk about what diseases those might be in a minute. But in terms of PKU, basically you can see here that if you've got a blood spot from a newborn that has a very high level of phenylalanine, you get bacterial growth around that plate. And so you want to check that patient out again with another blood spot and see if that phenylalanine is also elevated. Okay. And back in those days, you could sort of gauge the amount of phenylalanine you thought was in that blood spot based on a standard curve increasing from right to left of phenylalanine that was spiked into blood spots. That was then, this is now. So moving to now, this is a tandem mass spectrometer. So today, with the advent of very sophisticated analytical tools, we can screen for 10, 20, 25, upwards of 30 genetic diseases using tandem mass spectrometry. Okay? That has really been a boon for newborn screening, but it also has some challenges, which I'll touch on in a second. There are questions that still exist that are somewhat ethical questions and also sort of state specific mandates. And what do I mean by that? Different states screen for a different panel of genetic disorders. So one state, for example, Washington, might screen for 20 or 25 inborn errors. Wisconsin might only screen for 6 metabolic diseases. So you can imagine that there could be some tenseness, some rankling, if you had a child born in Wisconsin with a certain genetic disease that wasn't screened for in Wisconsin, but was screened for, let's say, in Washington or Idaho or California. And that's basically because each of the states still has autonomy for what their screening program is and what diseases they want to screen for. The whole idea that Guthrie developed for newborn screening was sort of threefold. You have to have a disease that is reasonably prevalent, and phenylketonuria was certainly that, 1 in 10,000. You have to have a treatment for that disease. In phenylketonuria, for example, you restrict phenylalanine in the diet, okay? You give enough so that the child can grow and you have normal brain development. You have to be able to have a system that can screen for it. And you can see here that even back in the 60s, they did have that. So therein lies sort of the conundrum. You can detect a lot of diseases today, but for many of them, there are no real good treatments. Having said that, you still have the opportunity for the family knowing what disorder they have. There's the opportunity for genetic counseling, so you can know you're at risk in the next pregnancy and other issues like that. Just to put it in perspective, the United Kingdom only screens for 2 diseases, phenylketonuria and a disease called MCAD, medium chain acyl-CoA dehydrogenase deficiency. Don't worry about what MCAD stands for. But they only screen for 2. And there may be that they're trying to get rid of even screening for MCAD in the UK. So that's how we look for them. Washington screens for about 20 to 25 disorders. 9.1 million tests were run in 2011. Approximately 167,000 specimens were analyzed from 85,000 newborns. And the cost for that was about $70 per newborn evaluated. If I, as a parent, if you as a parent, would 69 or 70 bucks, I want to do it. I want to know. So what are the traditional treatment approaches before we really get into what's sort of new on the horizon for different metabolic diseases? Most treatments, even today, except for selected diseases, are symptomatic. So if a patient has seizures, you treat the seizures. If a patient has an acid-base imbalance, okay, you treat the acid-base imbalance or the renal tubular acidosis because of the kidney malfunction. Basically, the old concept is that you restrict the offending compound. So in phenylketonuria, you give them a diet which is low in phenylalanine and low in protein, but still sufficient so that they can grow and their brain can develop, okay? Or you can supply with the necessary product that isn't made because of the genetic defect. In PKU, that's tyrosine, okay? You can give cofactors, and what are cofactors? Those are compounds in the body, vitamins sometimes, that help enzymes work more efficiently. For example, one of the cofactors in PKU is BH4, tetrahydrobiopterin. I won't quiz you on that. You can give that, and actually that has become the genesis of a treatment for PKU, which is a drug called sapropterin. You can give vitamins. There are defects in certain pathways in individuals that require vitamins, like biotin. And you have a defect in enzymes like holocarboxylase synthetase, where if you give enough biotin, the enzyme activity can go up somewhat, and enough to give a normal outcome for the individual. You can also give what's called conjugators, compounds that will complex with the toxic compounds in the body and excrete that, make that toxic compound into a non-toxic compound. And I'll show you some of those in a second. Glycine and carnitine are a couple of those. You can give dietary supplements, essential fatty acids, docosahexaenoic acid, which is basically an essential fatty acid, fish oil, minerals, antioxidants, because especially when you restrict dietary intakes, very strongly, you have to give back a lot of minerals and supplements. Okay. Those are the traditional treatments. And I thought what I would just show you is a slide of the 3 disorders that we are studying currently in the laboratory. And we study them in mice. And what does that mean? So basically what we have done and what others have done for the last decade, and it's become pretty much accepted practice, is if you know where the gene is that's defective in the human patient, you can go into mice and you can knock out that gene. And if you're lucky and the animal survives, not always, you can study the pathophysiology of the disease in the mouse, and you can actually look at new treatment approaches in that mouse. So I study 3 diseases currently in our laboratory. One is a defect in the metabolism of GABA, which is the major inhibitory neurotransmitter, and it's got a defect in that enzyme. The other is maple syrup urine disease, and that is a defect in branched-chain amino acid conversion. And basically, the branched-chain amino acids are isoleucine, leucine, and valine. No quiz, don't worry. And then I also study in mice PKU. We've gotten very interested in alternative treatments for PKU, and that's a block in the enzyme here, phenylalanine hydroxylase. So how have we treated these diseases in the past? And that's sort of shown here. The pathophysiology of these 3 diseases are quite different. This defect, since it's a defect in neurotransmitter metabolism, is a neurological disease, okay? It's rare. I've studied it for 30 years. We've got about 400 or 500 patients. But basically, the treatment for that has been symptomatic. Patients have seizures, you treat seizures. The patients have attention deficit hyperactivity disorder, you treat that with drugs. For MSUD and PKU, you treat by dietary restriction of either branched-chain amino acids or phenylalanine, okay? You give large neutral amino acid supplements, and what do I mean by that? Well, branched-chain amino acids and phenylalanine are a component of what we call the large neutral amino acids, including tryptophan, including methionine, and don't worry about that. But for those amino acids to get into the brain, they have to cross the blood-brain barrier, and they have to cross it on a transporter called the large neutral amino acid transporter. And when these compounds get high in the blood, they overwhelm this large neutral amino acid transporter such that they get preferentially moved into the brain, and other important large neutral amino acids don't get into the brain. And that's very important for both of these diseases because some of these large neutral amino acids have very important roles, especially tyrosine, which becomes dopamine, very important monoamine neurotransmitter for movement, and tryptophan, which is another large neutral amino acid, becomes serotonin. Hugely important monoamine neurotransmitter. So that's how we treated them, or how we still sort of treat them today. But what I want to get into in the last few slides is what are some of the new treatments for these diseases on the horizon. So we'll talk about transplant, liver, kidney, and heart, which have been going on for a long, long time. Touch a little bit on what's the process called Domino transplant, which is gaining a lot of play or notoriety, if you will, in the literature. Cell therapy, which my lab is very much interested in, especially liver cell therapy, and we'll get to that. Bone marrow transplant or hematopoietic stem cell transplants. Enzyme replacement, and that has become a huge, huge area, and that's really predominantly working on the lysosomal storage diseases, which I'll just barely touch upon. Enzyme therapy, and finally, of course, gene therapy and stem cells, just very briefly. And if I was going to talk about liver transplant, I have to acknowledge the 2 people that were really the fathers of orthotopic liver transplant, Sir Roy Calney and Dr. Thomas Starzl, who I got to know when I was at the University of Pittsburgh. And when I left Pittsburgh in 2009, he was still active in his mid-80s. He's really, and there's the Starzl Transplant Institute at Pitt. Not to throw you off a little bit, but on the bottom on the right is basically a liver for transplant and then kidney over here. And orthotopic liver transplant is really corrective, not curative, but corrective metabolically for many diseases that have a primarily liver phenotype, okay? And those would be diseases, for example, like the urea cycle disorder. That's where in your liver you have to deal with nitrogen from all sorts of metabolism, and you have to excrete nitrogen. And the urea cycle is a pathway that takes care of endogenous nitrogen. Porphyrias, which are diseases, different diseases, and glycogen storage diseases, but these are all primarily liver phenotypes. Okay, so orthotopic liver transplant is very good for that. What are the drawbacks with liver transplant? Well, graft versus host disease, chronic, if not lifelong, immunosuppression, okay? And especially today, the lack of donors, which has sort of burgeoned into cell treatment, and something I'll get into at the very end of the talk is humanized tissues. There are at least, I think the last time I looked at it in 2011, there were 17,000 patients awaiting liver transplant on the list. What are the pluses? Much improved quality of life for the recipients, if not life in general, okay? Metabolic control. So, for example, in maple syrup urine disease, you have long-term improvement in the branched-chain amino acid levels and improved somatic growth. So there are always pluses and minuses. So I mentioned Domino liver transplant. Now, what does that mean? And it's actually a very clever approach to trying to expand the use of the number of livers that are out there. Not exactly the right time for this at breakfast, but we'll just go on. So if you look here, a diagram of what a Domino liver transplant is this is the concept. You may have a cadaveric donor, deceased liver, that can go into a patient, for example, with maple syrup urine disease, okay, with classic disease. In the Mennonite population, there's a very, very high frequency of classic maple syrup urine disease. The outcome is dreadful, okay? Those patients are not going to do well, and they almost always go to transplant, and they go to the University of Pittsburgh for transplant, okay? You take the liver out of the MSUD patient, and you put it into a Domino recipient. So cadaveric, MSUD. In this case, just as an example, sclerosing cholangitis, okay, liver failure. This individual is going to die, okay? And so that individual is going to benefit even from receiving a maple syrup urine disease liver. That's going to save that individual and basically make him or her a chimeric individual. What do we mean by chimeric? Well, they've got MSUD disease in the liver, but in the muscle, in the kidney, in the heart, they have normal activity. So they may have a little bump in their branched-chain amino acids in blood because they can't very well metabolize them in the liver, but they're going to be alive. So it's a win-win in a way for patients. It's basically a mechanism to avoid end-stage liver disease for people that are very low on the transplant list, okay? But you have the same problems and the same negatives. You have graft versus host, you have very invasive surgery, and you have long-term immunosuppression. That's the payoff versus end-stage liver disease. Something that we've gotten very interested since my time at OHSU and my collaborations with Marcus Grompe is cell transplantation, especially hepatocyte transplantation. Okay, so we know that hepatocyte transplantation can work, and we know because mythology tells us it will work. Didn't get much of a chuckle. So if you remember your mythology, the story of Prometheus, who was one of the Titan gods, and he broke a rule with his brother Zeus. He gave fire to humans. And so what Zeus did to Prometheus was chain him to a rock, okay, and he had an eagle peck out his liver daily. It just tortured him to death. Unfortunately, the liver regrew. And so we know that liver regeneration will work, and we know it because of mythology. And it does, because the liver is the one organ that can regenerate in the body. It has certain transcriptional factors that it can go back into the cell cycle, basically, and turn itself on for regeneration. If you remove the a quarter, a half, up to 3/4 of the liver of a rat or a mouse, you can regenerate that liver, okay? And so that's burgeoned the idea long ago that we can transplant cells exogenously and get them to engraft into diseased liver and get them to expand. So what is hepatocyte transplantation? And these are basically hepatocytes shown over here. They make up about 90% of the liver mass. That's the hepatocyte is what does the activity in the liver. And the liver is probably my favorite organ next to the brain. It carries out anywhere from 400 to 600 different reactions, so be good to your liver. Okay. So far today, hepatocyte transplantation has become very valuable as a bridge for people that are in liver failure, as a bridge to get them, hold them along until they can get a transplanted liver. And that's usually been working in urea cycle diseases, a very pronounced liver disease. We and others have performed preclinically hepatocyte transplantation in a series of, in many murine models, and especially in maple syrup urine disease. And we just had a paper accepted in hepatology where we actually used human amniotic epithelial cells, which are basically sort of pluripotent stem cells that come from placenta, which is discarded at birth, so there's no ethical concerns there. And we took human placental amniotic cells and transplanted them into the liver of maple syrup urine disease mice. And they engrafted, and they expressed enzyme activity. So it's possible. The problem so far with hepatocyte transplantation for us and for others is giving the cells that are exogenously put into the liver an engraftment advantage over the endogenous cells and an expansion advantage. And that's been a challenge. And we're still working on different ways to get over those bumps in the road. Bone marrow transplant has been used for quite a few years, and it still has a lot of utility. The beauty of bone marrow transplant is that those cells can also cross into the blood-brain barrier. When you give a bolus of an enzyme for an enzyme replacement therapy to an individual, usually that enzyme will work peripherally, but it's a big, big protein. And that big protein usually doesn't get through the capillaries of what we call the blood-brain barrier. So bone marrow transplant has been used historically in blood disorders, hemophilia, factor VIII, severe combined immunodeficiency syndrome. It requires bone marrow ablation. You have to destroy the bone marrow of the individual before you put in new bone marrow. And one can imagine that's a dangerous process. So there's huge risks involved, okay? But you have to weigh the benefit of the risk of SCIDs versus bone marrow ablation. There's also graft versus host disease. There's risk of infection. And it's also used in a lot of the lysosomal storage diseases, bone marrow transplant, because the enzyme, as I alluded to a moment ago, can cross the blood-brain barrier, or the cells can cross the blood-brain barrier. And those are genetically what we call genetically competent cells. They have a full complement of the enzymes that might be missing in that individual. Enzyme replacement therapy. So probably I have one of the joys in my life is I'm a co-editor in chief of a journal for our European Society, the Journal of Inherited Metabolic Disease. And I would say 60% of the papers we get are on enzyme replacement therapy for lysosomal storage diseases. Gaucher disease, Fabry disease, now the mucopolysaccharidosis. This has really become a very, very big area in metabolic disease. And basically, why does it work and how does it work? So when we talk about lysosomal storage, we talk about enzymes that need to get into the lysosome. And it's an acidic little organelle in the body, like the Golgi or the ER, the endoplasmic reticulus. And basically, the lysosome degrades a lot of big macromolecules, just to keep it in most simplistic terms. The neat thing about enzyme replacement therapy is that for an enzyme to come from the nucleus to get processed in the Golgi and get into the lysosome, there's a docking mechanism. There's a receptor mechanism for that enzyme to get into the lysosome. And if the enzyme is properly conformed and synthesized, it can bind onto that receptor and get into the lysosome. And that is the whole idea of enzyme replacement therapy in lysosomal storage diseases. You give the patient genetically prepared enzyme that they're missing that is properly folded, that has all the receptor components on it. If you give it to them intravenously, it can find the lysosome and get into that lysosome and be functional. So it's really a beautiful concept, and it works, okay? The downside is it's exceptionally expensive. So usually treatment for some of these lysosomal storage diseases will run $200,000 to $300,000 a year, okay? You have to get boluses of enzymes, intravenous administration, perhaps every 2 to 4 weeks, okay? And the downside, again, is there's no neurological improvement. That enzyme, given peripherally, can't cross into the blood-brain barrier. The upside, of course, is a major quality of life and improvement in survival in some of these diseases. So it's a really burgeoning area, and the specificity of the lysosome and its structure is what makes enzyme replacement therapy work for lysosomal storage diseases. Enzyme therapy is really beginning to gain some traction, and this is not gene therapy, but it's enzyme therapy, and it's not for lysosomal storage diseases necessarily. But for example, I show it here. This enzyme, well, phenylalanine gets converted by an enzyme called, and you don't have to remember this, phenylalanine ammonia lyase to a non-toxic compound called tranexamic acid. It can be metabolized that way. So enzyme therapy for PKU is by giving a recombinant enzyme called phenylalanine ammonia lyase to patients to basically go into their system and break down any high level of phenylalanine, convert it to a non-toxic compound, and that's excreted, okay? That's also given intravenously. Enzyme therapy for PKU is right now in clinical trials, okay? It is expensive. There can be immune response, so you can have some rejection. But the benefit for a PKU patient is there's very much decreased dietary protein restriction, and that's a huge benefit. If one can imagine the stigma of an adolescent that is not allowed to eat, for example, a hamburger with their friends or pizza with their friends because they're on PKU diet, they're on very severe protein restriction. So the benefit of this is a very decreased protein restriction for those patients. Just about the last slide. And something I just wanted to touch upon, and no, you will not need to remember any of this, but I thought I'd just touch on it. One of the real hot areas today, and kind of gets me excited, is humanized tissues. So you've known for a long time that probably in Texas, for example, probably at the DeBakey Center, you could donate a pig heart and use that as a transplant in humans, okay? Today, we are already past the stage of humanizing mouse livers and now humanizing pig livers. So what does that mean? So I'll give you just a quick example. This is the tyrosine pathway, okay? And you don't have to worry about it. It's just an amino acid that gets converted down to metabolites in the body. There's a block down here in an enzyme called fumarate acetoacetate. Basically, it forms a disease called tyrosinemia type 1. Marcus Grompe at OHSU really was had the genesis and the idea of making a mouse knockout model of this human disease. Now, in this human disease, in tyrosinemia type 1, if you don't treat these patients with a drug called NTBC here, it blocks this enzyme, the dioxygenase. If you don't treat patients, they uniformly progress to hepatocellular carcinoma, and that's a very bad outcome. If you give them NTBC, it's basically life-preserving, okay? The same idea worked in the murine model. If you didn't give the knockout mouse model of tyrosinemia type 1 NTBC, they progressed to hepatocellular carcinoma. Give them NTBC, they're fine. So Grompe had the brilliant idea that you can take this knockout, have it on NTBC, remove NTBC, let the liver start to progress to carcinoma, and transplant exogenous normal cells, cells that didn't have tyrosinemia but had the normal complement of the enzyme fumarate acetoacetate, okay? And he utilized that cleverly as a mechanism for engraftment enhancement. The endogenous cells, because they carry the defective gene, die off. The exogenous cells are engrafted, and they expand. And he could convert more than 95% of that mouse liver to normal FAH, fumarole acetoacetate activity. And that's kind of burgeoned the idea of humanizing animal tissues, okay? Now individuals have already put human cells, and we've done this also in MSUDs in mice, transplanted human cells into mice and got good engraftment. And you can knock out this gene if you wish to and use that as a model. And they've done the same thing in pigs, so that you can actually take the pig hepatocytes and transplant in human hepatocytes and convert the majority of that liver into human hepatocytes, basically almost a human liver. What's the idea? Well, if you think about the limitations of orthotopic liver transplantation, here you're starting to be able to generate as many livers as you need for transplant. So it's becoming a huge field and a very exciting field. And as I alluded to a moment ago, with 17,000 individuals on the transplant list awaiting, having a source of a new and reproducible source of human livers is going to be a big boon on the treatment market. So gene therapy, of course, I don't do gene therapy. It's the holy grail. Because when you transplant a liver or you transplant cells, you are not curing, you are correcting, okay? You may normalize the liver phenotype, but you're not curing it, okay? Still in the nucleus, in the genetic material, you haven't put in the normal gene. And now, today, the real goal is to normalize or to cure through gene therapy by replacing the defective gene. Gene therapy really got a setback in the late 90s because of an attempt to give an adenoviral gene therapy vector to a patient with ornithine transcarbamylase deficiency, which is a urea cycle disorder. And the individual died. And that kind of put a hiatus on gene therapy in humans, but it's really coming back today. It's already common for most of the blood disorders, okay? And you can also use the idea of pluripotent stem cells. That's one of the approaches that we used in the MSUD mice, taking stem cells that can actually be used, transplanted, that can differentiate into a variety of cells, whether it be hepatocytes, whether it be blood cells, whether it be neuronal cells or glial cells, and you can directly inject those into the brain. And that has a beautiful potential for diseases like Parkinson's disease and Alzheimer's disease. So, wrapping it up. The future, gene therapy is going to happen for a lot of disorders. We just have to cautiously move through all the animal models. Animal organs are already used, and I think that humanized pig livers will be on the horizon fairly soon. The estimates are within the next 5 to 10 years. Individualized genomic medicine is here. I mean, the idea of having the complete genome, your complete genome, sequenced is here. It's present. And so specific therapies will be targeted to specific individuals. But I also wanted to point out before closing the emergence of the whole area of epigenetics. So we used to think in sort of the dark ages that genes were sort of stagnant. You had what you had. But now we clearly know that there's a lot of genetic modifications of genes in individuals over time with exposure to environmental insults, with exposures to toxins, with exposures to oxidative stress and different foods and carcinogens and things like that. Let me close it by acknowledging the people that give me money to do the research. NIH, the PKU Alliance funded our work. Many colleagues. Kara Vogel, who does a lot of the animal transplantation and mouse work in our laboratory. Gary Pollock and WSU Spokane for allowing me the opportunity to be here and to be a lot closer to my kids. And especially some special thanks to a couple of colleagues. I showed her first, my wife, and of course, my dog, who is my closest companion at times, especially after a rotten day in the office. The dog is always happy to see you. And let me open it up for any questions and say thank you for your attention.
Medical education research at WSU, we've coined it a new program, really. It's still formulating as WSU Medical Sciences. And we talk about WSU Medical Sciences because WSU has a real stake in what's happening in medical education. But the medical education enterprise and the medical school involved is actually the University of Washington's. So we're trying to define ourselves in this collaborative process as we grow and become more involved with more parts of the medical education pipeline. So I'm going to be using the terms medical education or medical sciences really at WSU, and much of that is in the context of the WWAMI program, which stands for the Distributed Regional Medical Education Program of the University of Washington, the Washington, Wyoming, Alaska, Montana, and Idaho, where the WWAMI acronym comes from. So I can't really launch into this without making sure we're all sort of in the same place in our understanding, and so bear with me if this is a complete repeat for you. But this is the medical education pipeline, and I don't have a laser on this, and my battery's about to go, but it's a long road to become a physician. Okay, you have to go to college at the beginning, and then you have to go to medical school, and we refer to medical school as undergraduate medical education. It's a 4-year program. It's confusing that we call it undergraduate because it's not like it's beyond college, but it's undergraduate as far as medicine's concerned. So we refer to it as UME, and it's typically composed of 2 years and 2 years. The first 2 years is somehow an assembly of basic science training with clinical influence on that training, so a clinically relevant basic science education. And then the following 2 years are a clinical immersion experience where students do rotations in different specialties: internal medicine, family practice, pediatrics, psychiatry, general surgery, and so forth, so that they emerge sort of as an undifferentiated physician, okay? And from there, they enter into their graduate medical education phase, or GME, and that involves, for everybody, residency, and for many, a fellowship experience after residency, which sort of hones their skills in a very specialized area of medicine. And really, this GME experience can be anywhere from 3 years, if it's just that's kind of the minimum length of a residency with no specialty training after that, to really up to 12, actually. If you do a 6-year residency and a 3-year fellowship, you're into it for 9 years, and that's actually very common. Some surgical specialties actually have up to 9 years of residency with a lot of specialty research involved, and then a fellowship after that. So you can do the math. Most physicians are not practicing until they're well into their 30s as an independent practitioner. So if you do something to change medical education, enlarge it, grow it, you don't see the effects for a decade or so after you've initiated that. So there's a lot of urgency around what we're doing here because we have very clear goals in terms of increasing our physician workforce and making things more robust in Spokane. But if you start now, you still have that lag. So what do we do here in Spokane? Well, this is really how the WWAMI system works. In Spokane, 2.5, almost 3 years ago now, we started a first-year basic science program. That constitutes the first year of medical education. This occurs here in Spokane. It's occurred for 38 years in Pullman. It occurs in Idaho. It occurs in all the WWAMI states. All the students in those distributed sites do a first-year program, and then after that, they all go to Seattle for year 2. And that's been the WWAMI model throughout. And so you can see as you grow in the region, you grow the second year in Seattle, and that's really a choke point for the system. So as we contemplate growing much bigger in Spokane, people in Seattle are saying, hey, wait a minute, if you put 40 or 50 or 60 or 70 more students in Spokane, that puts that many more students in Seattle in the second year, and we really can't handle that. So what we're talking about now is an expansion to take on this year 2 content and have students here in Spokane, potentially for all 4 years of their medical education. The other things that we've been doing for a long time is the clerkship offerings in Spokane. A student can really take all their rotations in the 3rd and 4th year in Spokane. We just have a capacity problem. We don't typically put that many students through rotations in Spokane. So our job there is to increase the capacity of those clerkships. And I put WSU and University of Washington the way I did because that's sort of how it may flesh out. WSU is very much responsible for the first year and will be very much responsible, I expect, for the second year when it comes. The University of Washington manages the curriculum and does the curriculum oversight for the 3rd and 4th year clerkships, and that may very well stay the same, although all these things are open for discussion. But here's where we're going. We had a feasibility study, so the University of Washington, Washington State, and community leaders in Spokane were all involved in a process of studying how we might expand this process to 120 students entering class size in Spokane, and a complete year 2 in Spokane, and growing those clerkships offerings so that this number of students could stay in Spokane and do all of their training. Now, we'll probably still have a movement of students because it's a distributed program, but those would be the numbers. So this growth is really one of the prime movers for growing the WWAMI program and, as a consequence, or a coupling, the biomedical sciences at WSU. So that's the point I wanted to make in those 3 slides. And this is how we're going to do it. We basically need more faculty, and we need more space. And as many of you know, we have a building that's been funded in the design phase, and we're hoping for construction funding for that, because without at least one new facility, we're not going to be able to accommodate even the first step of this growth. So with that said, let me just introduce you to the faculty that we hired as the first phase of this program. And the WWAMI program in Spokane is different than all the other first-year WWAMI programs around the region because we are a small campus with a small faculty, and the faculty we do have don't have that much strength in the basic sciences. So we actually had to hire, and the state appropriated funds for us to hire some of our own full-time faculty into the WWAMI program in Spokane. That's not been the model in the other university phase first-year programs. Those programs are set down on research-intensive campuses where they have lots of faculty already doing biochemistry and molecular biology and all these things, and so you just get teaching effort from those existing faculties. Here we're building it, and so that's a fundamental difference, and we want to build it further, and so that's what's going to create the change on this campus. So Dr. Roberts there was the first hire. He's a basic scientist. Ted Chauvin is a very recent hire on the research track. These 2 people work in the same area, okay? Because it's Dr. Roberts' talk today, he's going to give you 1 slide on what he does. Okay. I can talk longer than that, and there was 5 slides in here originally, but 5 slides would take me 50 minutes. Yeah, exactly. So here you go. We actually study the male reproductive tract, and we study a structure called the epididymis. So that's why I have to show you a slide, because when you say, what do you do? Well, we study the epididymis. Okay. Well, I didn't say I studied cancer, right? And everybody goes, oh, okay, you study cancer, or you study whatever. If you study the epididymis, you have to explain yourself. So we study the epididymis, and basically this is a structure that everybody knows about this structure here. This is the testis where spermatozoa are produced. But what most of you probably don't know is when a spermatozoa leaves the testis, it is incapable of fertilizing an oocyte, even if it encountered one, okay? Which is a very rare event for a spermatozoa, by the way. So when those cells leave the testis, they go into the structure called the epididymis, which just is a long tube that's supercoiled into this small space. And as they move from the top of the tube to the bottom of the tube, they magically acquire the ability to fertilize an oocyte. And they also acquire that ability to swim, both of which are required for fertilization. So a lot of male infertility and a lot of dysfunction in the male reproductive tract is tied to the dysfunction of this structure. It's also become a very important target recently for the development of ideas around contraceptive interventions on the male side, which have been lagging in recent times, say the last 50 years or so. So we spend our time investigating this structure. And one of the attractions for me coming back to WSU, besides coming home, was WSU is probably, if not the strongest, one of the strongest universities in the reproductive sciences in the world, and certainly in the country. We have 96 reproductive biologists between Pullman and the Moscow, Idaho campuses, and a Center for Reproductive Biology that I'm now a faculty member in that program, and working with some of the premier reproductive biologists there has been a real pleasure and attraction for me. Our second faculty that we hired was a cancer biologist out of Texas Women's University, and she studies telomeres. And telomeres are the ends of your chromosomes that need to be maintained and actually replicated if your cell is going to divide. So if you get a tumor, in order for that tumor cell to divide and to grow indefinitely, it has to have a transformation of the telomeres in order for that to happen. And so she studies the role of telomere maintenance in cell immortalization, and it has great for relevance to cancer. She's a fabulous molecular biologist. She's funded by the NIH, she's funded by the American Cancer Society, and she's going to do great things here in Spokane. So she had some spots on TV not too long ago. Some of you might have seen something about her. The next hire was Jonathan Weiser, a picture on the upper left there. who is all three of these individuals are sleep neuroscientists. And we are just down the hall from one of the premier sleep and performance research centers in the country. And this was one of the regions that we had such success in hiring neuroscientists into our program. Jonathan came first from the Stanford Research Institute, where he was very much becoming an established investigator in the pharmacogenomics of sleep. And he studies basically sleep and how drugs and different factors interact with that process. I'll say now, and I'll say it again in a moment, he's collaborating with Matt Layton. There's a lot of overlap between drug interactions, addictions, and so forth in the sleep process. Doctors Kappas on the right there and Sensormai on the left are both sleep scientists who are interested in the connection between sleep and metabolism. There's very much a connection between sleep disorders, metabolism, eating, and metabolic disorders as well. They're very interested in the endocrinology and the paraendocrinology of that process. And so between the three of them, they formed actually a key group within the WAMI program very early. Ava and Levente come to us most recently, just over a year ago. And then the other two people that are integrated into our system are people that we didn't hire as an overt external search, but we're here in Spokane, and we very quickly adopted them into our WAMI program. One, of course, is Dr. Belenke, who is the head, along with Hans von Dagen, of the Sleep Center just down the hall here, and really was the catalyst for us being able to attract our sleep neuroscientists. And Dr. Matt Layton, who's in the front here, who's the director of our psychiatry residency here in Spokane, the Spokane track, as well as our third-year clerkship in psychiatry, and also the first-year course director for systems and human behavior. So these people that I've just introduced really serve as the nucleus around which we are expanding education and research in the WAMI program. Now, we have many other people teaching in the WAMI program. Dr. Novan is one of those key point people who's doing that. But we have several others teaching other programs, and I didn't mention at the beginning, but we get a tremendous amount of help from faculty from Eastern Washington University, from some faculty in Pullman, and we draw them from the community as well. But our intent is to grow the WSU-based faculty to make this program really embedded in the university system. So as you're looking at what we do, and have already started, we have these key areas in sleep already. Because if you're going to ask the question, how are we going to grow out as we get a second year and begin to recruit more faculty and different things happen? What's the research enterprise going to look like within WSU Medical Sciences? And so you just sort of put up there what we're doing, and I'm going to talk about this in a moment, but Wei Han is our lone cancer biologist in the WAMI program, but she's not the lone cancer biologist in the WSU system. There are a number of people in the College of Pharmacy who are studying different aspects of cancer in the laboratory and more clinical settings, and as most of you know, the College of Pharmacy is going to be relocating to this campus as soon as we have a new facility, and that is going to greatly strengthen our cancer biology area. Reproduction is actually not an area I would think needs to be overtly strengthened in Spokane because it's already a tremendous strength of the WSU system, and we would actually have no trouble attracting other reproductive scientists to this campus if we want to, based on our existing strength. And then probably, in addition to sleep, one of the premier programs at WSU is in the School of Nursing with John Roll, who directs a very vigorous research program in mental health and addictions, and he collaborates closely with Dr. Matt Layton in that, and this is an area of tremendous potential within our biomedical sciences writ large. We also have a lot of efforts in research and really implementation research in diabetes and obesity, again in the College of Nursing and the College of Pharmacy, and so this is another area of strength, not directly in the medical sciences program that I'm talking about, but it exists. And then in the community, we have Kathy Tuttle, who's one of the premier researchers in the nation and internationally known for her work in chronic kidney disease. And interestingly enough, chronic kidney disease is one of the primary morbidities associated with diabetes. And so these things are all very much combined with each other. And then I would be remiss if I didn't mention informatics, which is a very important area for sort of doing clinical research and doing especially community-based clinical research in this enterprise. And again, not directly part of WSU Medical Sciences as it associates with the WAMI program, but a very important program. We have Dr. Duratha here this morning who's on the leading edge of that effort for WSU. And of course we have INHS, which is one of the premier institutions in informatics in the country. So when I look at this distribution of things that we do, we clearly have a strength in the neurosciences, and I see that would be an area that we could grow and become a really world top tier place for doing the neurosciences, particularly in the area of sleep. The other areas don't organize as well by subject matter, although they do, or they can be grouped in subject matters, but I'd like to just lump them together as efforts in cellular and molecular biology, because these people are doing things really at that level. But of course, these are not mutually exclusive. We have a lot of cellular and molecular work going on in the neurosciences as well, and so these circles will begin to blur, but I just want you to see sort of, as we contemplate how we want to grow, we need to grow in focused areas. And of course, the informatics and the diabetes will fit in there as well. Right now, there's sort of a delineation between what's going on. Most of this research is in behavioral or clinical research, basically not in the laboratory, and this over here is in the laboratory. But again, over time, I see a lot of mixing of those two things. Certainly a lot of diabetes research is conducted in the laboratory as well. So I guess what I'm trying to point out here is we're small now compared to large medical centers, but we have laid a very nice nucleus for growth in very important areas that will distinguish this campus if we grow them in strategic ways. And some of the drivers of that growth are going to be the expansion of the WWAMI program, which we've already talked about. It's going to be the consolidation of the pharmacy program in Spokane as well. And that's, this consolidation of pharmacy is not really new money into new programs. This is just a relocation of existing research and faculty and education processes to the Spokane campus. So that's going to have the effect in Spokane of immediately elevating our game. We will also experience strategic growth in our areas of strength that's not associated necessarily with programmatic increase. And I can already give you an example of that with our hiring of Drs. Weiser and Zensarmai and Kappas in connection with the Sleep and Performance Center here. Dr. Jim Kruger, who is probably the premier basic neuroscientist in the area of sleep, who had his research activities in Pullman, petitioned to be relocated to the Spokane campus. And we're in the process of, he's actually already here, and we're in the process of making the facilities available for the complete production of his research potential here in Spokane. He brought with him 4 research assistant professors, 2 graduate students, and 2 technicians, and some undergraduate researchers. And so it was really a shot in the arm in terms of our critical mass for basic research. And we think that as we grow these various nuclei of research strength, that we will attract other people on what we call soft money, grant monies, and so forth, to actually grow our programs that way as well.